Hey, what's up, everybody? As you can see in this particular recording, I am not allowing you to see me. That's because at the time of this recording, I haven't really put myself together this morning, haven't put on my face, haven't showered, haven't done my hair. So you don't want to see this version of Rogers. It might actually frighten you and upset your stomach, cause severe emotional trauma. So we're just going to go right to the lecture. And let's see. Okay, so what we're doing in this particular lecture is talking about something called the excitation contraction coupling. Now, if you remember from anatomy, we have three types of muscle tissue. We have skeletal, cardiac, and smooth muscle tissue. Now, when it comes to all of them, they are all excitable, meaning that they must be stimulated electrically in order to initiate the contraction process. And so we're focusing on skeletal muscle here. And so what we're looking at is a special type of synapse called the neuromuscular junction. Let me kind of just increase that a little bit there so you can see things a little bit better. There we go. And so we're looking at the neuromuscular junction. So we have the axon terminal of a somatic motor neuron. We have the muscle cell here. We can see the sarcolemma, which is the muscle's membrane. This specific area of the sarcolemma right here is called the motor end plate. And it's the wrinkly portion of it here are called junctional folds. And on the junctional folds, we can find the acetylcholine receptors. Now, that's where obviously acetylcholine will bind once it's released from the axon terminal of the motor neuron. So, like I said, the muscle must be stimulated electrically. And so this is where it all begins. The excitation of the excitation contraction coupling is the electrical stimulation. Now, this whole process is basically taking all of the events from the stimulation, the electrical portion of it, initiating a series of chemical events, mainly acetylcholine, and then the chemical part of that will ultimately translate into the mechanical portion, which we would then see as movement. So, and this all happens during a special period of time called the latent period, L-A-T-E-N-T, latent period. Now, here's where it starts. So the action potential shows up at the axon terminal, causes the release of acetylcholine, excuse me, and the acetylcholine will diffuse across the cleft where it's going to bind with receptors here on the junctional folds at the motor end plate. And as we can see here, it's going to open up those receptors and form an EPSP. Sodium comes in, potassium leaves. That depolarizes the membrane to threshold, and then boom, we get an action potential. Now, the actual depolarization to threshold has a special name, which we can see right down here. It is called the end plate potential. Now, don't worry about numbers in terms of voltages or threshold or anything like that for the muscle content here. We're not going to cover that. All you need to know is that the action potential forms. And once it forms at this spot, okay, it's important to remember when we compare this type of action potential to the action potential in the neuron, one of the bigger differences is how it propagates. If you remember from our lectures on the neuromaterial, when an action potential forms at the axon hillock in a neuron, it only propagates unidirectionally. So it goes from the hillock to the axon terminals. But here on the muscle cell, when we get the action potential here at the neuromuscular junction, it spreads omnidirectionally. It spreads out in all directions along the sarcolemma. Now, let's get to what happens next. I'm not too concerned that you guys know how it propagates, because when we look at a slide like this, it's very similar to the action potential. When the membrane depolarizes, it's because of sodium channels opening up and sodium coming in, and then it repolarizes by those sodium channels closing, potassium channels opening, and potassium leaving. So that's exactly how it happened in the neuron. Okay, so let's get to this slide here. Now, when the action potential is spreading along the sarcolemma, it's going to get to a point on the sarcolemma called a T-tubule or a transverse tubule. Why the T-tubule is special is that it allows, or it helps conduct the action potential from the perimeter of the sarcolemma on the outside of the cell, and it conducts it inward, takes it down into the cell. Now, what's a little hard to see in this picture is that on the T-tubule side of this membrane, we have what are called voltage-sensitive proteins embedded in the membrane that are physically linked to calcium release channels on the other side of the membrane that are embedded in the wall of what are called the terminal cisterns. So you have one terminal cistern here on this side, one on this side of the T-tubule. The three parts together, 1, 2, 3, are called a triad. Now the terminal cisterns are part of a larger structure called the sarcoplasmic reticulum. And its only job whether it's the larger portion of the SR or the actual cisterns here, their job is to store calcium. Okay, so when the action potential comes down the T-tubule and it hits those voltage-sensitive proteins, they will change shape. And since they are physically linked to the calcium release channels on the cistern side, those channels will open up and calcium will flood the cell. Now, calcium is represented in this picture by these red dots. So as you can see, they flood the cell. And then calcium's only job in this entire process is to undo the blocking action of tropomyosin. Okay, so let's just kind of review real quick what tropomyosin is and how it's why it's important. So let's go to this slide real quick. Now, there's four main proteins. There's more than four when it comes to the anatomy of a muscle cell. But there's typically only four that I want you to remember, four that I emphasize. We have myosin over here, the thick filament. Here's an individual myosin molecule. And then over here, we have actin, the thin filament. Now, actin is made up of individual actin subunits with myosin binding sites on them. And so you have two strands of actin subunits kind of twisted over each other. And this is where troponin and tropomyosin are involved. They help make up, we can call it if we want to call it the actin complex, because there's three proteins here. So actin is one of them. Now, tropomyosin is represented as this long, thin strand-like protein. Troponin is this little yellow globular protein here. Now, what's kind of hard to see in these pictures is that troponin and tropomyosin are physically connected to each other. They are linked. Now, at rest, the tropomyosin, when the muscle is at rest, the tropomyosin covers up the myosin binding sites on the actin subunits. And that's why we call troponin and tropomyosin the regulatory proteins, whereas actin and myosin are the contractile proteins. And so what calcium does is calcium will come along. Let me get back to this one. So calcium comes along, binds to troponin, and that physical union between the two causes troponin to change shape. And since it's linked to the tropomyosin, it pulls the tropomyosin out of the way. And like we can see here, it exposes the myosin binding sites on the actin. And until that happens, myosin cannot bind with actin to form a cross bridge. And we need a cross bridge to form in order for the muscle to actually contract. So that's why calcium is so important. And that's when I say it undoes the blocking action of tropomyosin, that's exactly what I mean. It helps move tropomyosin out of the way and exposes the myosin binding sites. Okay? And we, believe it or not, we haven't even contracted yet. But let's actually get to that. So let's go over here to something called the cross bridge cycle. Now, I don't ever start at number 1. I usually start kind of right here. And so I look at it as, if you were to kind of print here, let me give you a suggestion. Print this slide as a page by itself, And then I would say print this slide as a page by itself. This one, or you can probably go with this one here. I would probably go with this one since it's a little bit bigger. And then print this one here. And line them up in order, 1, 2, 3. Okay, line them up linearly, 1, 2, 3. And talk your way through the process. And so imagine we're coming at it from this perspective. We're coming along the pages over here. And then this is our last page, and this is the first spot we get to, if with our eyes when we're looking over the pages. So the myosin head here, in order to form a cross bridge, which is the physical link between actin and myosin, the myosin must be energized. And in order to energize it and kind of move up into a cocked position here, ATP has to bind to it and then get hydrolyzed. So when we're looking at myosin right here, we would describe it as being in its high energy state or cocked position because ATP has been hydrolyzed. Now it has a high affinity, AFF entity, for actin, and it has a strong attraction. It wants to bind. And so now what happens is that head will move up, and like we can see here, it forms a cross bridge. Now here's what you want to remember. Myosin binds to actin. Actin does not bind to myosin. It doesn't work that way. So the cross bridge is formed. Okay, the cross bridge is formed. And then we go this direction, just like the arrows indicate. Now, look at this picture here at the top. Look at this picture here on the right. And generally when I do this lecture face-to-face, I'll tell everybody, take a minute or so, look at these two pictures, this one, and compare them. And there's 3 things that I want you to notice, 3 big differences. And so, like I said, I'll give everybody a minute or so to think about it. But here are the 3 major differences. You notice that there's arrows right here that indicate movement, indicate the actin sliding over the myosin. We see here that the ADP and the phosphate detach. And then we see, look at the myosin head here versus here. It's cocked. It pulls. Now, I didn't list them in the necessary order. So here's the actual order of importance. So the ADP and the phosphate detach from the myosin. So we can see they're attached here and they detach. This initiates what's called the power stroke or the working stroke. And as you can see, the myosin head is in a unique position here. But once the ADP and the phosphate detach, the myosin head pivots and it pulls on the actin. And the actin, according to the arrows, slides this way over the myosin, and the sarcomere shortens. So again, it goes in that order. Number 1, the ADP and the phosphate detach. Number 2, the power stroke occurs, which is the myosin head pivoting and pulling, and the actin slides over the myosin. It's called the sliding filament model of contraction. And again, it goes in that order. There's no variation. And it's important to note that it's not just one power stroke that makes a muscle contract. It's kind of an oscillating movement. And so you have to use your imagination where one myosin head pivots and pulls, and then another one pivots and pulls, and then another one pivots and pulls, and so on. You get my drift. Okay? Now, that makes a contraction. That makes a full-blown contraction. Now, in order to relax, we see something happen here. We see ATP come along, and ATP will bind with the myosin head, breaking the cross bridge, like we see here. The myosin head drops back down. Okay, now let me go back up here for a second. Once the myosin head pivots and pulls, excuse me, it enters what they describe as its low energy state. Okay, so it's energized over here. It does the job here, and then it's tired, essentially. It's in its low energy state. So we've contracted. Now, in order to initiate, like I said, relaxation, ATP has to break the cross bridge. Okay? Now look at this little thing here. This is kind of an interesting note. In the absence of ATP, myosin heads will not detach, causing rigor mortis. As you know, that only happens one time in our existence, after we die. And so whatever position the body is in when ATP is gone and the cells experience death and stop producing ATP, that's the position that the body will remain in unless forcibly manipulated, which is kind of a not a pleasant thought. But that's, if you've ever wondered what rigor mortis is and what causes it, it's because of a lack of ATP. Okay, so we're starting to relax now. And what I like to tell students is that when we start to discuss relaxation, what I want you to do is imagine moving backwards. Okay, and what I mean by that is let's go backwards in the slides and reverse the steps that we used to get to this point here. Okay, so we go backwards. So let's go back to this point here. Now, backwards. Calcium detaches from troponin. Okay, troponin goes back to its original shape, like we can see here. When that happens, it pulls and moves the tropomyosin back to its pre-stimulated configuration, which is covering up the myosin binding sites. Okay, and then the last thing we want to address is what happens to calcium. So calcium gets actively transported back into these cisterns. And that's it. That's the entire process. But the last thing we want to mention as we wrap this up is, why is ATP so important? Okay, when you've been watching this video, a muscle cannot contract or relax, okay, with two main things, ATP and calcium. Now, let's go back actually real quick to the cross-bridge cycle slide. Now, ATP, there's three big reasons we need ATP, so make sure you write this down because this is very, very testable material. So number one, right here, to energize the myosin head. Okay, without ATP, without the hydrolysis of ATP, there's no energized myosin head, and with no energized myosin head, we don't get a cross-bridge. With no cross bridge, there's no contraction. Okay, so that's number one, to energize the myosin head. Number two is to break the cross bridge. That's the second reason we need ATP. Without ATP, a muscle cannot contract. Without ATP, a muscle cannot relax. Okay, so number one and number two. And then lastly, we go back to this slide. And the third main reason we need ATP is to actively transport calcium back into the cisterns. And so I mean, think about it. We can't contract, we can't relax any skeletal muscle without ATP. It's physiologically impossible. All right, that's actually it, my friends. You know, we're approaching the 18-minute mark. I think this has got to be a record for me to do this particular lecture in. Normally, I'm much more long-winded and take an hour or so to do this. But anyway, if you have any questions, please let me know. Hold on. Let me just transition here into, we just talked about the reasons we need ATP. And that's kind of a good transition point into the second muscle lecture where we talk about how the body replenishes ATP. All right, so again, you've got to remember the three main reasons, and then that will help the second lecture, the second muscle lecture, make a lot more sense. All right, so if you guys have any questions, you know how to find me, and have a good day.